So welcome to our new LIBREhub seminar for this semester. And today we have as a guest Dr. Leonel Malacrida. He's currently at Universidad de la República and Institut Pasteur of Montevideo. He's an associate professor and principal investigator at the Advanced Bioimaging Unit. And so this is very exciting because he has been a leader in the development of imaging tools in Latin America, and also our colleague in the through different I think multiple funding instances through Trans-Zuckerberg initiative. So welcome, Leonel, and we're very excited to learn today about the FLIM data analysis using PhasorPy.
Thank you very much, Vicente, for the kind introduction, and also for the invitation. It's my great pleasure to be participating in these meetings that I often participate as a listener, and I also go to the YouTube channel many times. So congratulations for the success on the meeting. So can you see my screen now? Is okay?
Yes, we can see your presentation.
Okay, so as Vicente mentioned, I will try to show you a little bit about the motivation of my research with the fluorescent lifetime imaging microscope and hyperspectral imaging, and I will try to convince you how valuable it can be to move into the Phasor approach for the analysis of the FLIM and hyperspectral imaging, and I will try to show a little bit of the application that we are given to the Phasor analysis for dermatology, and then I will talk a little bit about the PhasorPy, which is a new initiative that we are working in terms of software to provide, an open source code that can handle with the FLIM and hyperspectral data through the Phasors. And then I will try to wrap up showing some of the opportunities we have here in the Advanced Bioimaging Unit for the region to access to these technologies. So to start, I would like to say why to bother with fluorescent lifetime? Well, fluorescent lifetime is a characteristic for any fluorophore that is independent of the concentration, and it gives you an idea of the average decay that it has a fluorophore in some environment. This makes a very, very special quantitative approach to measure, in cells or in living, animals. And one important thing about the fluorescent lifetime is also can be affected for many different situations like ions concentration, pH, hydrophobic properties, oxygen concentration, energy transfer. So it can be a ruler, a tool to measure other things that can happen in a living system. So the lifetime, as I show here in these two examples, can give you a lot of insights of what is going on in terms of a membrane organization. And this is a cell labeled with a night red, and this is the lifetime measurement, and this is a biosensors using FRET to understand the macropinocytosis, etc. So fluorescent lifetime is a really powerful tool to quantitatively approach microscopy. But one of the main reasons for why I believe, my opinion is not that widespread, is because it's rather complicated and it's difficult to analyze the data. The time of exposure you have in your system is always limited. So basically, you will collect a few hundreds of photons per pixel, which give you a profile of pixel histogram like this, instead of the classical view of the decay that we have. And this makes very tough and difficulty when you want to do analysis, such as fitting with mono-exponential decay, double exponential decay, or even more complicated. The other big challenge is that all these methods of fitting is time-consuming and computing-consuming, and this requires expertise. So basically, you need to become, in some way or another, expert in the field in order to take full advantages of the possibilities and the capabilities that FLIM does. So the last thing that I want to mention, FLIM instrument are most of the time customized microscope. There are only few examples nowadays. Leica has released a microscope that is full capable of FLIM by default. So you order a microscope that will do FLIM. Some of the others are always add-ons to an existing microscope, and this makes always complicated. So the question is if we can do something simple for experts, and I will try to convince that the Phasor is the way. So, but before to go to the Phasor, I also want to show a little bit of the potential of the hyperspectral imaging. Hyperspectral imaging is all now available in almost any commercial microscope. What it does is basically you take a stack of X and Y, and then in the T dimension, you have lambda. So you have many pictures as this with different, ranges of the spectra that you are collecting. And using the right, tools, you can end up having a confocal image with the 3 or 4, whatever channels you have. There are different ways of approaching this. You can have a single detector and you can collect different steps of the spectra. You can have, an array of a detector, and you can collect all the range in a single shot, or you can use this technique that was developed in the place where I did my postdoc, in the Laboratory for Fluorescence Dynamics with Enrico Graton, using the sine and cosine filters, and we will see a little bit more later on. But again, when you want to analyze the data, the challenge is you need to use, some of these tools like supervised classification analysis, principal component, or linear mixing, in which you need to know some things about the sample you have. You need to know what are the components you have. You need to know what is the shape. So you can feed all this information in your analysis, and then you can obtain the information. So far, basically, again, you need to have an expertise on the sample and on the method that you are using to take a full advantage of the spectrum. So what we propose to use is the Phasor plots. And the Phasor plot is not new. It's something that started with the definition of the Phasor transformation in the 1981 by Weber. And the introduction of this plot, which is this semicircle that we call universal cycle, we will see why it's called universal, was done by Jason, Hall, and Graton in '84 to work with cuvettes. And since the beginning, this transformation, this is the Fourier transformation, normalized Fourier transformation, enables you to put basically the fluorescent lifetime in a vector that give you basically information about what is going on in terms of your sample. If you have a single exponential, if you have a multi-exponential, if you have a reaction in the excited state, etc. So what are the main advantages? Well, you don't need a knowledge a priori because if you look at the Phasor, you will realize what is going on. So the only thing that you need is to learn this new language. The other thing is the rules are very simple and it's model-free. You don't need to fit, which is in a way an important thing because you are not assuming a priori if you have one component, two component, or autofluorescent or whatever. So if you are interested in going a little bit deep in this, I wrote this commentary some year ago, something like that, where I try to emphasize the power of the Phasor plot because you can go to lifetime, you can go to spectra, and you can do many quantitative measurements using the spectroscopy of this method for this purpose. But how the Phasor plot is done? Well, the Phasor plot, if you take in the lifetime, right? If you take a decay, you will have many decays pixel by pixel, and then you do this transformation, right? You do the mathematics, basically the computer does this, and then you start to obtain from each of the pixel the position of this vector, which is the G, the X axis, and the S, the Y axis, for each of the pixels you have here. After all, the representation in the Phasor is a cluster, which is basically a way of representing the density of pixels you have. As higher the density, it's redder, the position here, and then you analyze this data. The calculation is very simple. Basically, you multiply the decay by a cosine or sine function, and then you integrate this. And when you do this integration, you see you will get the real and imaginary component, and it will lie in a point on this universal cycle when we're speaking of a single exponential decay. So this is the first definition. Every time that we have single exponential decay, no matter what, if the system is calibrated, should fall on this universal circle. So by looking at the data, you immediately will realize if you have a single exponential decay or not. The other thing is if you have a longer lifetime, right? As we can see here, this red line, well, when we do the calculation, this will show up later on the anti-clockwise movement of the vector. And again, if it's a single exponential decay, will be on top of the universal cycle. If not, will be in some other place. So now we can see that just by looking at the phasor, learning these two simple rules, you will immediately realize two things. First, well, these two are single exponential decay. Second, the lifetime of the black is shorter than the lifetime of the red because the position is toward to 0, 0, while this one is toward to 1, 0. So by looking again at the phasor without any mathematics, you can tell the change of lifetime. So the other important rules are the linear combination. So we can take advantage of the vector properties on the Fourier space. And if you have a position in the center inside of the universal cycle, this could be because of the linear combination of two lifetimes. If you have the lifetime tau 1, which is longer than the tau 2, the fraction of each of the component is basically the trace of the counterpart that you have in the linear combination of these two components. So basically, by doing some simple geometric mathematics, you can obtain these two fractions and then quantify how much weight you have of the lifetime 1 and the lifetime 2. The other important thing is that you can also see things which are outside of the universal circle. And this happens when you have reaction during the excited states, when you have a delay on the emitting of photons. So by, again, looking at the phasor, you will immediately notice if you have something going on, and then you can study what is this delta phase and how you can interpret this delta phase. There are many examples of things like the excimer formation, reaction in excited state as energy transfer during FRET, if you look at the acceptor, or if you are looking at dipolar relaxation by the solvent. The last property, which is very valuable of the phasor approach, is basically the reciprocity principle, which means that if we have a fluorescent lifetime imaging or a spectral imaging, and you transform it to the phasor, you will get clusters here. And using a simple tool we call cursors, these circles, you can select a region of interest here, and then you can trace back where are those pixels in your image. So as you can see, this start to be very important for the imaging, because we have hundreds, if not millions and millions of pixels that we can have information about the heterogeneity on the lifetime distribution you have. But also you can do the other direction, which is you can also make region of interest in yourself and then look at the scatter that you get in the phasor, which tells you that the lifetime inside of the cytoplasm or the nucleus in this cartoon example can be heterogeneous and can give you variabilities within the sample that you are studying. And again, I want to emphasize that you can measure, if you have the few component, you can measure how much fraction you have of one and another, and you can obtain quantitative information very easily. The last thing is, in the introduction, is about the spectral phasor. Well, just modifying a little bit the phasor transformation, where we include the lambda, we can obtain a phasor transformation for 3 spectra. I simulate these 3 Gaussian as a spectra, for instance, and you can see that the maximus is changing, but also the width is changing. And for the narrow bluer spectra, the position will come up here, the red here, the green here, the green here, and the red here. So you can see immediately that as the spectra shift, the position will lie on a revolution counterclockwise, depending on how far in red you are going in the spectra. And the second thing that you would realize is that as narrow is the spectra, more long is the modulation, so the vector is farther on the radius and broader closer to the center. So again, looking at the phasor, you can also see if there are changes in the spectra without any assumption or fitting or nothing. And again, the linear combination rules is very valuable because you can see if you have a point here, which has combination of these two components, will lie in the middle, the same for this, the same for this. And again, it's also true for even after 3 component, 4 component, 5 component. So the Phasor is really valuable tool to analyzing, data in terms of hyperspectral image and lifetime imaging microscopy. Yes, again, we can transform the data to the Phasor, but then we can go also back and color map using these cursors. And this is a sort of a 2x2 simulated image in where we have 4 pixels with 4 different combination of each of the spectra. So the first example that I want to show you is a project that was led by Sofia Martinez, dermatologist, Bruno Schutti, biomedical engineer here in the Advanced Bioimaging unit. Both worked together in this project in which we wanted to understand if some of these tools can be valuable for the melanoma diagnosis. Melanoma is a very bad problem in the world because it can is the second most important cancer in Uruguay after lung, and this follow more or less the statistic in the world. It can be very lethal because 30% of the diagnostic annually die because after diagnosis, because either the diagnosis is wrong or is too late. And we rely nowadays basically in 2 different approaches in order to understand if you have or not a melanoma. Well, first of all is this presumptive clinical diagnostic in where the models are evaluated using this ABCDE rule, which they look at the asymmetry, the border, the color, the diameter, and evolving. And a physician with the dermatoscope need to look at your skin and see if these changes, capture his attention, and decided to remove this region of the skin because it's a presumptive melanoma. After that, you need to go to an anatomopathologist, and basically, based on hematoscilin and eosin, you need to discriminate between these 2. One of these is melanoma, one of these is regular nevus, so a benign lesion. And this is basically, again, is work of the pathology, the anatomopathology, who look at the cut, at the slide, and based on their knowledge, they learn how to recognize these pathognomonic features in the tissue, which make always difficult the diagnosis, because unfortunately we don't have such a molecular marker for the melanoma provide robust information for the dermatologist. So with this, the motivation of our work was if we can propose something that can be simple and accessible for many of the clinics around the world, in which we can take advantage of the biopsy that's most of the in the melanoma diagnostic and procedure. Basically, the physician remove a biopsy and then send this to the dermatopathology, and the dermatopathology study the hematoxylin and eosin. Nowadays, there are coming some new ideas using multiphoton microscopy to deep penetrate in the skin, and they look at the free and bound NADH and FAD concentration. And there are some very nice preliminary work on this. We are also working in this way. But the story that I want to tell you today is going in the direction where we will try to use the things that are more simple and accessible in the lab. So the drawback of going to the multiphoton FLIM is you need to have a very expensive microscope, and you need to have experts on the microscopy in the hospital to handle with the data. So we propose this. We propose to have slides without labeling embedded in paraffin. So basically, we don't modify at all the procedures of the dermatologist. And then we do basically Phasor transformation of the autofluorescent we have here using just UV light, 405 excitation in a confocal microscope capable of doing spectral imaging. The challenges are that, you know, this is a small piece of a lesion that requires, you know, 8 tiles by 14 tiles. Each of these is 1,000,000 pixels, so 1,000 by 1,000. So it's a colossal amount of data, 32 channels in C. And this is a small, you know, it's like most of the time you have samples we can be a centimeter big. So this was the first point where we have to manage to develop our own library. And I will speak a little bit more about the PhasorPy, which is something that is open for the community and everybody can take advantage of it. So showing now in direct into the results, you know, this is the hematoxin eosine, and this is the counterplot images of the autofluorescent. These are non-labeled. This is basically 2 different slides. One is stained, one is not. And this is the image that we obtain when we excite with 405 the same samples. And you see that you start to see some features here, but it's hard to discriminate where is which. Nevertheless, if we use just a color map in which we highlight the change on the phase, so how much is changing the center of mass of the spectra that we have here in the Phasor, you can immediately realize that, yes, we can separate this area, which has something weird there. And this is basically the region of the melanoma. Then we decided that, okay, how we can provide some numbers to this. So for me, that I am not an expert in dermatology, at least these tell me that there is something weird there. But there is any way that we can quantify this? Well, the answer is that we have decided to make region of interest based on the know-how of the dermatology, and then analyze again the cluster that we obtained for the nevus and for the melanoma, and measure basically the shift in the angle and the shift in the modulation. So we generate a histogram of the modulation and the phase for all the pixels that we obtain in each of the cases. And you can see in these two, you immediately notice that there is a shift in the phase and modulation that can capture these differences quantitatively. With that, we decided to analyze 5 lesions of nevus, so these are benign lesions, and then another 5 with a melanoma diagnosis with different Breslow. This is how deep the melanoma has penetrated in the tissue. And these are the histogram for all of them. We quantify this histogram, calculating the center of mass of the histogram, so we can now do, you know, easy statistics. And you see that the center of mass of the modulation and either the phase can capture this difference between the 2 set of samples. And then we end up deciding to make a scatter plot in which we plot the phase versus the modulation. And we realize that this enables us to have like 2 clusters in which we can see that the interval of confidence is very good. So we can easily split between, you know, the 2 situations, melanoma and nevus, which is the first attempt that we generated in order to provide quantitative results to a dermatologist. But then after that, we wanted to understand what was exactly this autofluorescent, from where was coming from this autofluorescent. And one interesting thing of the Phasor plot is the linear combination. So if we can have, you know, pure collagens, we bought these collagens in, you know, some provider, chemical provider, and then we also speculated that we can have NADH, elastin, melanin, and FAD. Well, we can obtain the spectra for each of the different components and then plotted them in our Phasor. And after that, play around with the linear combination to see if our data is aligned or not with this component. And as you can see here, all the data is in a way within this quadrangle that we generate here. And we can have now an explanation of which are the components that explain, you know, the difference between the melanoma and the nevus in the composition of the molecular fingerprints, as we like to show. So we are using the molecular fingerprints of the tissue as a tool to determine if this is a melanoma or an nevus. So then we wanted to see if this has differences specifically between the two sets of samples, and we made high resolution imaging. This before everything was obtained with a 20x 0.75 numerical aperture objective. So this is a more coarse imaging, and here we use 64x 1.4 oil immersion objective, so we can have, as you can see, more high resolution. And when we look at the composition that we have in the sample, we start to understand that the fluorescence from the nevus is mainly related to collagens, FAD, and elastin, while in the melanoma, there is Y more fat, and this is the reason for why this cluster of data is more pulled to this position. There is also melanin here, and there is a component that we cannot identify, but we speculated that can be a different, oxidative state of the melanin, because there are different oxidation of melanin, and we are now working on understanding this. We are trying to produce this melanin in a synthetic way to see if we can give it a name to this composition here. But in any case, we were, successful in determining, the component and the role of these different things. So this is the story that I want to tell about how Phasor can be powerful for, the dermatology. It's interesting that we were trying to figure it out. It is very easy, hyperspectral imaging UV without a commercial microscope, nothing sophisticated or custom-made, can be valuable for the dermatology. What we are working now, the direction is, well, we have, hundreds and hundreds of slides, so this takes a lot of time because we need to do all these tests. So we are trying to come up with a more, reliable method, and we are trying now to implement some custom-made microscope using these sine cosine filters. We want to compare this also with multiphoton imaging, so eventually compare what information gives each of the methods, and then we want to combine all this information with new scores and also with protocols of artificial intelligence. To approach the other part of the talk, I want to present this PhasorPy project. PhasorPy project started in 2021 with the thesis of Bruno, when he started with this project, and he needed to put together this code in order to handle with all the data, because it was impossible to do it before with the SimFCS software developed by Enrico Graton that we were using for more than a decade. So this software basically started with the funding from Chan Zuckerberg in the Open Science program. And we proposed to develop a Python library based on the open software principles, and try to basically make advantage of all the functionalities of SimFCS, but now in this open source. So we are able to handle with FLIM data, hyperspectral imaging, and we will do phasors, histogram, cursors, threat analysis, calculation of other variables that we can take advantage of the Phasor upload, but also a mixing, clustering, segmentation, etc. This is a software that aims to be built as a community, as a collaborative open source. It's stick to the MIT licenses, so it's very, very permissive. And we are working in many of these things as I will show you now. So the team is basically nowadays, myself, Enrico, who was former PI, now Michelle has this role. Michelle Dingman from UCI has the role in UCI. Bruno Sciuti and Bruno Panunzio are both part of the Advanced Bioimaging unit and are working in this project. And Christopher Cole, which is a senior programmer, is leading the programming in California, working together with the Brunos. So this is the web page, so you can go to phasorpy.org, and all the documentation is here. If you want to download any of the code, it's in our repository in GitHub, so you can go there, explore take advantage of all this, as I mentioned, is MIT licenses. You can take full advantage of it. And I want to highlight a couple of things. We are making a huge effort in order to be capable of opening all different kind of files from many different brands. For instance, you know, SIM-FCS data, Becker and Hickel, ISS file, OMETID file, PICO-Quant, and Leica, because we can also export from Leica data to PICO-Quant. And we are now trying to manage to also open other sources like OMETID, SAIS, Nikon. And so if there is anyone who has some of these instruments, we are very need for data that can be useful for the project. And I will show you how you can contribute with the project, not only with the code, but also with the material. The other important that I want to highlight is also the functions. We are implemented a very large group of functions, and we will keep updating this function. I want to capture your attention in two, which is the Phasor transformation. Traditionally, we use the FFAT by SciPy, but now we develop our own Phasor transformation because using Cyton, Christopher Gold managed to improve, you know, the speed of the Phasor transformation in an order of magnitude, and this can be very, very important when we think in very large set of data like the tile that I show you, or 3D stack of time data. And this was part of the innovation that we are proposing with this PhasorPy. You can go to the page and you have many different tutorials. If you go inside, for instance, to the fluorescent resonance energy transfer, you will see all the functions, all the documentation, and there is an interactive blog that you can download so you can take advantage of the, you know, a priori understand if you have, you know, a set of fluoro for a donor, how this will play around with the frequency, the lifetime of the donor, the lifetime of the receptor, you know, if you have autofluorescence, bleeding through, etc. All these variables that a priori give you an insight before even to jump into an experiment. So the last thing that I want to mention is, well, how you can contribute. Yes, you can contribute with code. Of course, it's absolutely welcome. We have very, you know, very strict rule in the code writing. So we basically, you can go and understand what are these rules. But yes, everyone is welcome to contribute with code, also with documentation. But also, I would like to highlight that we also are welcome to propose enhancement. You know, if you can think that there is something that can be better, please make a pull request, and we will try to improve it. The shared data, this is very important, and I want to highlight that we have a PhasorPy.org project in Zenodo, so you can deposit your data, and this is very welcome. And also you can propose, you know, report backs or etc. So this is part of the Advanced Bioimaging unit, which is the place, the center that we are leading here in Uruguay, who has basically this fantastic thing that support these 4 pillars: services, training, dissemination, technology-driven research, and biological-driven research. And I want to just highlight a couple of things that are connected with the Phasors and with the capability of, you know, taking advantage of Phasor. Annually, we have this Advanced Bioimaging workshop, always in the 4th week of November. We are already working in the 6th edition, and we cover all these topics, but also others, FCS related. We also cover deep tissue imaging, single plane illumination, super resolution. So if there is someone interested, please reach me, and I will be happy to provide this information. All the lectures, a priori lectures, are deposited in our YouTube channel, so you can go there. There is more than 120 lectures, and you can take advantage of all this material that is already there. We developed this project, which is basically the train the trainer, in which people from core facilities in the region can attend to the workshop, but then they stay for another 10 days. And the idea is that every year we focus on a technique. Past 2 years, we focus on hyperspectral image and Phasor analysis and expansion microscopy. And already we have like 20 imaging scientists from all over Latin America, Uruguay, Peru, Mexico, Argentina, Brazil, and Chile, who has taken advantage of this. And for me, it's a fantastic tool that we generate, because now we have a network of colleagues with know-how on these tools that most of the time are already available in the places, but they are not taking full advantages because the lack of the expertise with the Phasor approach and the handling with the data. This year, we will focus on FLIM and Phasors. So I would like to reach any of you who will have a FLIM machine in your lab and wanted to learn more about how to take advantage with the Phasor. Please contact me, and we will try to support to come to our workshop and to our training. Finally, I want to say that we also try to overcome the obstacles of approach or coming to our center to take advantages, and have this first experience with advanced bioimaging tool fellowships that we launched past year, and we will be opening new call soon, probably in June, July, for another round of 10 fellowships. And these are the ones that are already, happen. These are 2 stories that you can also visit our YouTube channel from Mexico and from Argentina, where they are taking advantage of the hyperspectral imaging for different purposes. These are the tools that we have here. 2 custom microscope, FLIM multiphoton, another machine for spectral and FLIM using fiber optics. We just have a microscope donated by Claire Brown. It's a single photon FLIM machine using a Pico One system that will be installed soon in the hospital. And this is a fantastic tool and an opportunity that Claire opened for all of us, also for training, which is very important for us. This, I want to invite all of you to our next Weber meeting, which is a meeting that we organize every 3 years with Enrico Raton and Dave Chason. Past edition was here in Punta del Este. Vicente was attending to the meeting, and we will have it again in Genoa next year, June, between 15 and 20. So mark in your calendar, and you are all invited to this fantastic conference that we always enjoy with our family. So with that, I want to conclude that basically the Phasor has removed the many obstacles to approach FLIM and hyperspectral imaging. It's really, a tool, power tool, quantitative approach that can, work with the mixing on the FLIM and the hyperspectral data without previous knowledge. And considering that this is a very, straightforward method, you can, very easily learn how to take full advantage. This is some literature that we have produced over the year. So I want to finish saying that Phasor is cool, and I hope I convince you that you can jump into this new space. And I want to finish acknowledging the team in the Advanced Bioimaging unit. These are the different channels that you can reach us. And thanks, of course, to all the funders, in particular Chan Zuckerberg and Latin American Bioimaging, who has been tremendous collaborators in order to promote the the accessibility of our tools to the region. Thank you very much.